Okay, everybody, welcome to the video lecture for Chapter 3, or actually what's likely to be the first of two, possibly three video lectures for this chapter. In Chapter 3, we're going to talk about some well-known and very common, in many cases, patterns that we see in the way that evolution plays out. There are about 10 or a dozen recognizable patterns, and once we get past some preliminary stuff here in this chapter, we're going to start hitting on those various patterns and talking about them in a little more detail. First thing we need to talk about, though, in this chapter is the relationship between taxonomic classification and evolutionary history. So in previous chapters, I said that basically, if we do our classifications correctly, they will sort of automatically show us the evolutionary history of the group of organisms that we're trying to classify. And the important point there is that taxonomic classifications, who goes into what phylum, class, order, family, genus, etc., those classifications should reflect evolutionary history. In other words, we should put organisms or species into a particular taxon, like a class or an order, for example, because they share an evolutionary history. They come from the same common ancestor. So if two organisms did evolve rather from the same common ancestor, then they will share many characters in common, because just like two siblings that came from the same set of biological parents, and those siblings will have things in common, two species that descended from the same common ancestor will have things in common with each other and with that common ancestor. And modern classification is based on shared characters, so it will reveal evolutionary history if we do it correctly. All right, so there are two sort of obvious processes that we can see in terms of the way that species are produced, or the way that species evolve. The first one is called cladogenesis. Cladogenesis is separation of a single lineage into two or more descendant lineages. So let's say, for example, we have an evolutionary tree that looks something like this. And we have, something like that, where we have an original ancestor at 0 that evolves into species 1, 2, 3, 4, and 5. That branching pattern, where you get multiple species from a common ancestor, that is cladogenesis. All right, separation of a single lineage into two or more descendant lineages. The other process, the other way through which species can evolve, is called anagenesis. Now, in anagenesis, you don't have multiple species evolving from a single common ancestor. What you have is an ancestor species that over time evolves into a descendant species. So this is change accumulating, over long periods of time, that's sort of transforming species A into species B. And you can see this sort of thing happening in the fossil record. Of the two, I think that cladogenesis is the more common form that we see, the more common pattern that we see. So anagenesis then is just directional change within a lineage. All right, now we need to talk about different kinds of taxa. Remember, taxa are classification categories. So we have kingdom, phylum, class, order, family, genus, and species, for example. Those are taxonomic categories, but the specific names of each, like a specific kingdom or a specific phylum, like, phylum Chordata, for example, those are taxa themselves. They are taxa. Taxa is more specific. Taxa fall within taxonomic categories, which are more general. All right, so we have three different types of taxa. First, we have taxa that are referred to as monophyletic. A monophyletic taxon represents an ancestor and all of its descendant species. Okay, it's an ancestor and all of its descendant species. So, for example, I could say that this portion of this tree here is a monophyletic taxon because it includes species 1 and 2 and their common ancestor. So it's that ancestor and its descendant species. That could be a class, or an order, or a family, or a genus. All right, that's how we want to do things. That's a proper taxon. Now, we also have two possibilities that we need to look out for. One called polyphyletic. So a polyphyletic taxa or taxon is when you have unrelated taxa that are descended from two or more ancestors. I'm going to show you some figures of this here in just a moment to make this more clear. So in polyphyletic, you have unrelated taxa that have descended from two or more common ancestors, but they're grouped into the same taxon, like the same class. All right, then we have, finally, what's called paraphyletic taxa. This is a taxon that includes some, but not all, descendants of a single common ancestor. All right, this may sound confusing, so let's look at this next figure here to try and clarify. Okay, first of all, let me just say that monophyletic taxa are considered to be the only valid type of taxa. They are what we're shooting for when we group organisms together. So again, in a monophyletic taxon, which is what's outlined here, okay, we have species A, B, and C all grouped into the same class or order or family or genus, let's say, because they all evolved from the same common ancestor. That's the proper way to do it. All right. Secondly, we have paraphyletic. This is not what we want. Okay, a paraphyletic taxon includes some but not all the descendants of a single common ancestor. So notice that we have species D, E, and F that do share the same common ancestor, but only D and E were included in that class or that order or that family. F was not included. Why not? Well, I'll give you an example. So class Reptilia, or what used to be class Reptilia, traditionally and for decades actually, has included the lizards, the snakes, the crocodilians, and the turtles. All right, consider those as like species D and E here. Well, recently it's been realized that birds are reptiles, but birds were in their own separate class, class Aves. So with birds not included in class Reptilia, even though they are in fact reptiles, class Reptilia that you see here, not having included birds, was considered a paraphyletic taxon once this was recognized. So as a result of that, class Reptilia no longer exists. It's being reformulated and reworked to now include the birds. And I'm not sure if they've decided on a name for the new class or not, but class Reptilia is a classic example of this sort of thing. All right, finally, polyphyletic. Again, also not what we want. This is a taxon that includes descendants of two or more ancestors, both of which have descendants that are not included in the taxon. So let's consider species H, I, and J a taxon, right? Like a class or an order, let's say. Well, H has this more recent common ancestor, I and J share this common ancestor, and so that right there means that they're grouped together, but they have different common ancestors, and then why not include K and G, right? That makes no sense. So this is a polyphyletic. It's a taxonomic category that poly has more than one common ancestor. That's also not correct. You wouldn't call that a class. What you might call a class is everything here, G through K. Group them all together in one class, and you see that their common ancestor is now here. All right, so as I said, monophyletic taxa are what we're looking for. They are considered to be valid taxa in the modern sense. And so, today we're looking at classifications based more on genetics, DNA sequences, and that sort of thing when you can. Can't do that for fossil species, obviously, but you can with extant or living species. Classifications in the past, especially prior to the 1980s, were based almost entirely on morphology, physical appearance, similarities and differences in plumage or digit number or you name it, whatever physical characteristic. Well, those things can be ambiguous and misleading and open to interpretation. So once they started to include genetic information, which is not ambiguous, DNA sequences are what they are, not open for interpretation, taxonomists began to realize that there were many taxa that had been produced and relied upon for a long time that were either paraphyletic or polyphyletic. So now they're going back and revamping all of these things based on new evidence and trying to develop exclusively monophyletic taxa. This is one reason why trying to teach taxonomy is so frustrating, because it changes all the time. It's being revised fairly heavily. Okay. So starting in the 1960s, thereabouts, there was sort of a shift in focus in terms of developing classifications, what we call taxonomy. And that shift was toward what we call cladistics. So cladistics, also known as phylogenetic systematics, is the modern method for classifying organisms. Cladistics focuses again on monophyletic taxa. So a monophyletic taxon like this one here, this grouping A, B, and C, that is referred to as a clade, because we come to those conclusions about those taxa using cladistics. All right, so monophyletic taxa are clades, and this is a result of a shift in emphasis back in the 1960s to one of common ancestry, grouping organisms together based on common ancestry rather than thinking about it in terms of just physical similarity alone. Sometimes physical similarity can lead you down the wrong path. A classic example of that is Linnaeus originally, back in the 1700s when he was developing the system, included bats in with the same group as primates. Why? Because they both had teats. They both had nipples. Well, we know that bats and primates aren't similar as a result of that. They both develop nipples independently, and so that's an example of a taxon being produced incorrectly as a result of looking just at physical features alone. Something had evolved 2 times independently, the teats, and that led Linnaeus to the wrong conclusion. He wasn't thinking about it in terms of common descent, getting features from a common ancestor, in other words. All right. Another important concept here, a couple of definitions or terms anyway, are what we call crown groups and stem groups. So when you look at an evolutionary tree, a branching pattern, right, a phylogeny as they're called, what you'll see is that within a taxon, a class, or an order, or whatever, you'll have species that have many ancestral features, older features, and some species that have newer or more derived features. The species that have the more ancestral features tend to be fossil species, although not always, and they are what we call the stem groups. Okay, the stem groups tend to have more ancestral features. So these are fossil species often that are more closely related to the crown group than to any other, but they lack the crown group's derived characteristics. The crown group are species descended from the last common ancestor of all living members of a clade. Another way of putting that is that the crown groups are species within a taxon that have newer or more derived features. Stem groups have more ancestral features. Stem groups are closer to the stem or the base of the branching pattern, the tree itself. Crown groups are more like the twigs at the very tips of the branches. Okay, so good example of that. Birds are a crown group relative to the stem group of theropod dinosaurs that we know that they share the same common ancestor with. Birds are in the same lineage as the theropod dinosaurs, but modern birds do possess many derived features. You can see that here. So here we have modern birds at the bottom, okay, and these are extant, currently are still living. But then we have lots of extinct species of earlier sort of proto-birds and then full-on dinosaurs like, the velociraptors, tyrannosaurs, that kind of thing. So these guys having much more ancestral features of the theropod group, these are stem groups, okay? Modern birds do have some features in common with theropod dinosaurs, skeletal features in particular, but they also have a lot of newer, more derived features that the theropod dinosaurs didn't have, like the asymmetrical flight feathers that produce lift, versus the symmetrical flight feathers of some of these guys here, these feathered dinosaurs that maybe were just gliders, could glide from one tree to the next, that sort of thing. Not powered flyers like modern birds because they didn't have the right kind of feather. The new design of the feather is one of the things that makes modern birds a crown group for this entire group of organisms, all again sharing the same common ancestor. So these guys, the stem groups, have many ancestral features. Crown groups have many newer features. Okay. All right. Now, whenever we classify organisms, what we're doing is creating a phylogeny, like what you see right here. This is a phylogeny, an evolutionary tree. It's a hypothesis about what we think happened in terms of the sequence of evolutionary change that produced all of these species from a common ancestor. Phylogenies are hypotheses. If I were to tell you that this might represent a class or an order or a family, let's say, okay, what I'm telling you is that that taxon is a hypothesis. And the branching pattern that we see within it is also hypothetical. This is what we think happened, but we just don't know for sure. Okay, there's lots of evidence supporting this, but we will never say absolutely definitively this is exactly what happened, because that's not how scientists operate. We always leave the door open for changing our ideas, okay, about things if new information comes along. So when you create these phylogenies like this, when you create a classification, what you're doing is looking at character states. So some species have this character, some species don't, right? So presence, absence, or various forms of a character or a feature. You're looking at character states, comparing between organisms, and then mapping those onto an evolutionary tree like this, a phylogenetic tree, using parsimony. Now, we talked about parsimony in the last chapter. Parsimony is, the process whereby if you are confronted with multiple hypotheses or explanations for something, you pick the one that's the simplest, because that one's most likely. That's parsimony. And that's the logical or deductive reasoning process that we use to reconstruct the evolutionary history here when we weren't around to see it. All right. So as an example of that, let's say we're talking about the development of opposable toes. So orangutans, gorillas, and chimpanzees all have opposable big toes. Humans obviously do not. So what we have here are two possible phylogenies, two possible evolutionary trees, classifications for what may have happened in terms of the evolution of the non-opposable toe. All right, look at possibility number one here. Under this possibility, we have chimpanzees and humans having the same common ancestor. And we pretty much know that this is probably the case because there's very little sequence divergence in terms of DNA between chimpanzees and humans. We share like 96 or 97% of our DNA in common. Humans and gorillas and humans and orangutans have many more differences in their DNA, so we must have shared common ancestors with them further back in time. Okay, so under this scenario here on the left, we have the evolution of non-opposable toes here. So because it came after the common ancestor with gorillas and orangutans, we're assuming that, or this says that gorillas and orangutans have opposable toes. But now we see it develops here before the ancestor 3, common to chimpanzees and humans, which would explain why humans do not have opposable toes, right? Non-opposable toes evolved here. It's in the pipeline. Humans therefore don't have it. But chimpanzees do, and they share a common ancestor after the development of non-opposable toes. That tells us that chimpanzees must have re-evolved opposable toes. Now that can happen. You can have a similar or the same feature evolve multiple times. All right, but let's look at this possibility. Here, we have the same ancestor for chimps and humans, but now we have the evolution of non-opposable toes happening after humans and chimpanzees split from their common ancestor. That would explain why humans have it, and it does not require chimpanzees to re-evolve opposable toes. It only requires one character state change, whereas this scenario requires 2 character state changes. That means that this one on the right is the more parsimonious evolutionary tree. That is the more parsimonious hypothesis, and of the two, that's the one that's most likely to be correct. Doesn't mean it is, just means it's the one that's most likely to be correct. It might well be that this is how it played out in terms of development of the opposable toe or non-opposable toe. But if we don't know, we pick the simpler scenario. All right, now let's talk about patterns of evolutionary change. So we're going to start getting into these patterns of evolutionary change, and I'm going to sort of number these as we go through so you know exactly what they are and can discern that from everything else. All right, the first pattern is that modern characters, so number one, modern characters evolve from pre-existing characters. All right, modern characters evolve from pre-existing characters. All right, this brings us to a really important concept in terms of understanding evolutionary history and constructing evolutionary trees, and that is the concept of homologous structures. Homologous structures. Homologous structures are structures or features in modern species that have been inherited from equivalent features in their ancestors. We say that a structure that two species share because they got it from their same common ancestor, we say that that structure is homologous. And a great example of this, and this is just one of many examples, is forearm structure in vertebrates, tetrapods, so four-limbed vertebrates. Okay, if you compare the four-limb structure of an early amphibian to that of a human, you see it's basically the same thing. In humans, you have the upper arm bone, the humerus, middle bones, or lower arm bones, radius and ulna, wrist bones, carpals, long bones of the palm, metacarpals, and then the phalanges, the fingers. Early amphibian, dating back to about 360 million years ago, exactly the same skeletal structure. In fact, if you compare it to all vertebrates today, or even extinct, that have four limbs, you see exactly the same structure. There can be lots of variation in form, but all the bones are typically there, or they're fused together in some different way, but the basic structure is there. Take a look at this next example. So here we have adapted for swimming. The flipper of a porpoise, humerus, radius, ulna, carpals, metacarpals, phalanges. Look at adapted for running. Horse, the forelimb of a horse, front leg, humerus, radius, ulna, carpals, metacarpal, phalanges. Well, actually one phalange. So in a horse, they've lost all but the middle finger, and they've lost or had some fusion of the metacarpal bones into one, and so this makes a much stronger skeletal support, supporting the weight of the animal for running long distances and very high speed. Here you see development of bones or bones sort of taking on different forms to result in a flat paddle-like appendage, a flipper, adapted for swimming. But the bone structure, the basic vertebrate forearm bone structure is there. Look at adaptation for flight. Look at a modern bird. Humerus, radius, ulna, carpals, metacarpals, phalanges. Loss of some digits, some fusing of bones because you need a very sturdy support structure to make the wing, right? That's the wing. Look at a bat. Humerus, radius, ulna fused into one. Carpals, very, very long digits for supporting the patagium, the skin of the wing that spreads between the fingers, forming the flight surface. An extinct pterodactyl. Humerus, radius, ulna, so on down the line. A long fourth finger, ring finger basically is what it is, that extends the wing outward and catches the patagium, the skin that the animal will use as a flight surface. And in fact, if you compare, the 35 or so thousand species of vertebrates today that have four limbs, and all those fossil species, now you're talking about hundreds of thousands, potentially millions of species, they all have exactly the same structure, the same bone structure. So the idea is that either Either that structure evolved hundreds of thousands or millions of times independently in all those different species of vertebrates, or that bone structure, forearm bone structure, was present in the common ancestor to all vertebrates, and it was just passed along, descended to them. Which of those two is the more simple explanation, the more reasonable? It evolved in a common ancestor who then passed it on to all its descendant species. That's the most parsimonious explanation. This is why we include all these vertebrates in the same group. They all have a vertebral column as well. That's another homology. DNA. Most all living species today have DNA. That's another homology suggesting common ancestry. So homology is a really powerful tool for reconstructing evolutionary trees. Okay, so last thing I want to talk about real quick before we end this particular video lecture is how it is that we identify homology. Well, the problem here is that sometimes you have structures that look like they're homologies when they really aren't. Okay, so think about, for example, the wing of an insect and the wing of a bat. Well, they basically do the same thing. There are some similarities in terms of their structure, but turns out they are a result of two separate independent evolutionary events producing wings. It happened one time in the ancestor of insects, flighted insects, happened one time in the ancestor to bats. There's no common ancestor between bats and insects that had flight ability, possess wings, so the fact that they both have wings is not a result of common descent. It's a result of at least two separate evolutionary innovations of wing structure. Okay, so how do we determine then if a structure is a homology? Well, first of all, we can look for correspondence of position. So where in relation to other structures is a character found? Look at the humerus bone. In every case, you find it in the same place. Look at the metacarpals. In every case, or the carpals rather, you find them in the same place. That is correspondence of position. If something is consistently found in the same place in relation to other structures across a wide swath of other species or different species, that's an indication it's a homology. All right, correspondence of structure. What parts is the structure composed of? Here we see it, right? Humerus, radius, ulna. Humerus, radius, ulna. Same thing. That's correspondence of structure, okay? That suggests that this structure is a homology because it's not very likely for it to evolve exactly the same way in the same sequence, humerus, radius, ulna, multiple times independently. Doesn't mean it can't, it's just not very likely. Finally, in terms of identifying homologies, we ask ourselves, when mapped on an evolutionary tree, such as the development of opposable toes that I talked about in the previous example, when mapped on an evolutionary tree, does its distribution on that tree suggest that it has been inherited from a common ancestor? That's another way to look at this, okay? So these three things then, correspondence of position, correspondence of structure, and their distribution on evolutionary trees can all be used as lines of evidence telling us that something may in fact be a homology, or conversely, telling us that it's not. All right. Okay, so I think we're going to stop there for this video lecture, and I'll pick up with some of the processes that can lead us to think that structures are homologous when in fact they're not at the beginning of the next video lecture.